plant disease management and sustainable agriculture.
Good afternoon, respected dignitaries. I am Professor Geeta, working as Professor and Head, Department of Botany, Bhadra University, Coimbatore. My research topic is biogenic nanoparticles for plant disease management and sustainable agriculture. So morning session we have heard a lot about nanotechnology and applications from Dr. K.S. Subramaniam talk. So now here I want to share with you the basic technology, basic things only, just synthesis, characterization, how these various types of nanoparticles used in agriculture include not only agriculture, human welfare also. I have concept first. Nowadays I am doing some of the work related to plant disease management. Just I want to share with you. So this is the content of my topic, that is nanoparticles, types of nanoparticles, advantages and disadvantages of nanoparticles, methods of synthesis of nanoparticles, then characterization of nanoparticles, activities of nanoparticles in plant disease management, and the key applications in agriculture, proposed mechanism of nanoparticles to mitigate biotic stress in plants, then type of nanoparticles used in plant disease management, and conclusion. These are my publications. I have done research on nanoparticles, synthesis and characterization and application for the past 10 years. Recently, we published one paper with the help, along with Dr. Sendil Raja. Here, we made chitosan nanoparticles loaded dimethoxycurcumin, and we applied these nanoparticles against Phytophthora infestans to evaluate the antibacterial activity of potato leaf blight disease. So, what is nanoparticles? So, we know very well food security and sustainability are seriously threatened in the world because of the increasing world population. It is estimated that the world population will be increasing up to 10 billion by 2050. Yield of crops are affected due to different biotic stresses, but which they could be effectively managed by application of chemical methods, but these methods leads to toxicity to human as well as environment. As a result, we need some innovative methods. Nanotechnology is one of the innovative science. Approximately 16% global agricultural production lost taking place due to phytopathogens, fungi only accounting for about 70 to 80% of the yield loss. So, green nanotechnology, it means synthesis of nanoparticles from plant pods, is an eco-friendly technology. It is widely accepted in agriculture. It refers to utilization of nanoscale materials, that is 1 to 100 nanometer, synthesized from living organisms. Nanoparticles, there are three major types. One is, it is classified based on composition they made: organic, inorganic, and carbon based. Organic nanoparticles made from lipids, proteins, and carbon. They have own advantages and disadvantages. For example, organic nanoparticles are often non-toxic and biodegradable, but they are sensitive to thermal and electromagnetic radiation. Inorganic nanoparticles made from silica, gold, silver, some metal oxide also. They show highly organized atom arrangement and also show more stability in nature. Some special unique properties like antibacterial activity, antifungal activity. Carbon based nanoparticles examples include quantum dot, fluorines, graphene. They show distinct mechanical, chemical, and physical properties. So, there are two common strategies for synthesis of nanoparticles. One is from top-down method, another one is called bottom-up method. In top-down method, nanoparticles are synthesized from bulk materials. In bottom-up method, these nanoparticles are synthesized from atoms, small particles. Under top-down method, physical method is coming. In physical methods, we can prepare nanoparticles using sophisticated instruments like ultrasonication, laser ablation, ball milling, sputtering. In bottom-up method, chemical methods as well as biological methods are coming. In chemical methods, we have to use chemical reduction and electrochemical process. Biological methods, we can use plant, fungi, even bacteria, mediated synthesis. This method is highly stability and biocompatibility of nanoparticles we can synthesize. So once nanoparticle synthesis in the lab, then we have to go for confirmation of the nanoparticle synthesis. Initially, when we add the plant extract along with the precursor solution, immediately color change will be observed. So not only color change will not confirm the formation of nanoparticles, therefore we have to go for various basic as well as advanced spectroscopic characterization. First one is UV visible spectrum. If you analyze the nanoparticle solution under UV visible spectroscopy, you can get the SBR band formation, which is not absent in the precursor solution. So it confirms the formation of the nanoparticles. Next is transmission electron microscope. Here you can find out the exact size and morphology of the nanoparticles. XRD analysis, you can find out the size and the crystallinity of the nanoparticles synthesized. Scanning electron microscope will tell the surface morphology of the nanoparticles. Dynamic light scattering will say the size distribution and dispersity of the nanoparticles. Atomic force microscopy will give the three-dimensional structure of the nanoparticles. FTS spectroscopy will give idea about the functional groups present on the nano solution. So this characterization is very important before applying these nanoparticles for various purpose. So what are the activities of nanoparticles in plant disease management? Nanoparticles have many properties like nematocidal activity, insecticidal activity, fungicidal activity, and activation of plant defense mechanism, bactericidal activity, and antiviral activity. There are three key applications in nanotechnology. Morning sir discussed, K.S. Subramanian has discussed. They can be used as nanopesticide, nanofertilizer, and a nanosensor. Silica and silver are mainly used as they act as insecticide because they will disturb the exoskeletons of the insects. Next, nano fertilizers. Some examples: nano urea, nano DAP, nano potash, nano apatite. Nano urea contain liquid form of nitrogen, nano DAP contain phosphorus, nano potash contain potassium, nano apatite contain both calcium and phosphorus. Then nano sensors, that is, it will with the help of nano sensor we can do precision agriculture or smart agriculture. We can monitor disease and insects, pollutants, soil nutrients, and water contents and temperature. Everything we can control. We can control from the real-time monitoring basis, and also we can assess the crop growth and yield. So what is the proposed mechanism of the nanoparticles to mitigate the biotic stress in plants means nanoparticles act on plants in combination of direct and indirect activity. Direct means they show direct antimicrobial activity. In this, nanoparticles, whenever attaches with the plant cell, they will disturb the cell wall and cell membrane and leads to death of the insects. The indirect means here these nanoparticles once binds with the cell membrane, it will enhance the cell membrane of the cell membrane and cell wall of the plant cell, means it will induce the immunity of the plant. So automatically plant will withstand the particular stress condition. So this is the antibacterial mechanism of the silver nano. For example, I am telling here antibacterial mechanism of the silver nanoparticles. If you apply the silver nanoparticles in a liquid form to the plant, suppose if you give spray, means first it will disturb the cell wall and membrane of the insects. Then second, they will once enter the inside, they will attach it to the ribosomes and make the denature. Third one, they interrupt the ATP production. Fourth one, they disturb the membrane and also forms lot of reactive oxygen species. This reactive oxygen species interfere with the DNA replication. Then finally, membrane denaturation is taking place. Then all the cellular things coming out of the cell, it leads to death of the insects. Then type of the nanoparticles used for plant disease management so far: silver nanoparticles, copper nanoparticles, chitose nanoparticles, zinc nanoparticles. But as silver, copper, zinc, they are somewhat metal-based nanoparticles. Sometimes morning we discussed about toxicity problems. Still now we don't have any regulatory frameworks using these nanoparticles large level to field conditions. That's why alternatively we are preferring to use chitosan-based nanoparticles, because chitosan is derived from the chitin of the insects. So from this chitin we are processing and we are getting the chitosan. This is biocompatible, biodegradable, eco-friendly manner. That's why comparing to other these four nanoparticles, we are using only chitosan nanoparticles for our work. So this is the silver nanoparticle. This is the silver nanoparticles. This is prepared from various from lower plants to higher plants. We can synthesize. This is the basic mechanism of the nanoparticles. For any nanoparticle synthesizing, the following this method only. Suppose if you take plant leaf, from this leaf you have to make the leaf extract. Maybe aqueous extract you can take it. After that you have to mix with the that is precursor solution. It means silver nitrate is the precursor solution. If you mix means automatically some color change will be taking place. It indicates the indicates the formation of the biosynthesis of silver nanoparticles. So here why it happens means plant extract contains some phytocompounds. These phytocompounds like phenol, flavonoids, terpenoids, tannins. These phytocompounds acting as capping as well as reducing agents. What is reducing agents? That is for example silver nitrate solution I am mixing with plant extract means the silver ion converted into the silver nanoparticles. This conversion is known as a reduction division. This is happened due to the action of the phytochemicals. Next, once nanoparticles are synthesized, they aggregate together and begin to grow. That process is known as nucleation. Then automatically they have to withstand for some time, at least for 3 months, 6 months. It has to stable. So in order to give this stable, these phytochemicals, some of the phytochemicals again encloses these nanoparticles. That process is known as capping. Okay, this is the methodology mechanism of the synthesis of any nanoparticles. So like this, we can prepare the copper nanoparticles. We can prepare the zinc oxide nanoparticles. So suppose if you evaluate any plant disease management, for example, one of the studies, we applied the biosynthesis zinc oxide nanoparticles to control the mycelial growth of the aspergil niger of the aqueous extract of the Symbobogon citatus, that is lemongrass extract we have taken. From this, we synthesize the nanoparticles. Various concentration we have used. Out of these concentrations, 200 microgram per ml zinc oxide nanoparticles showed less growth of the mycelium. So I already told you, among these silver, zinc and copper, we prefer only chitosan nanoparticles because chitosan is a sugar that comes from the outer skeleton of the shellfish, including crab, lobster and shrimp. Chitosan is derived from chitin through the process called deacetylation. It means which remove the N-acetyl groups and also to create the free amino groups. These amino groups will easily attach with any phytocompound or any phytoextract. That's why we are encapsulating, we are loading the phytocompound or phytoextract onto chitosan nanoparticles. Because of this property only, it will show the adsorption property. So this is a single method of preparation of chitosan nanoparticles. Here ionic gel method is used because first initially we have to get the chitosan solution. It is commercially available. So once we have to get chitosan, we have to dissolve in small volume of the 1% acetic acid, glacial acetic acid to dissolve properly. Then we have to add the cross-linking agent that is called TPP, trisodium polyphosphate. This is the chitosan itself is a negatively that is positively charged molecule. TPP is the negatively charged molecule. So once we mix both the things means automatically cross-linking will be taking place. They attach each other due to electrostatic interactions. As a result, we are getting the chitosan nanoparticles. Once chitosan we obtained, we have to go for washing, thorough washing with the help of water. 2 minutes, okay. Water, then we have to go for centrifugation. We have to filter it, we have to dry it and store it properly. Whenever we want, we have to dissolve in small volume of DMSO. Then in water, we will get the fine without any precipitation solution. This is also same with the as similar to the nanoparticle mechanism only. It is also following. So here the chitosan, now once chitosan nanoparticles are prepared, I want, because one chitosan nanoparticles did not show any specific activity. When you attaches any plant extract or plant phytocompound means it will act as a carrier. It will go and bring the and deliver the drug to the particular region only, specific region. That's why we are loading the phytocompounds or plant extracts onto chitosan nanoparticles. So conclusion, morning we already discussed, there are a lot of toxicity problems using nanoparticles. That's why we stopped synthesize these metal and metal-based nanoparticles. I am concentrating only on chitosan-based nanoparticles only. Metal-based nanoparticles, suppose if you use, before use in large scale means we have to go for more research, and also we have to sanitize the optical application and long-term environmental impact also we have to study properly. That's all.
Thank you, madam, for your nice narrative on the basics of nanoparticles and their characterization and their uses. Thank you very much.
Okay.
Now the paper is open for discussion, please.
Ma'am, thank you very much for a good presentation. In fact, before I started Nanotechnology Center in TNAU, we learned nanotech from you only. Whatever the slide you shown, 15 years ago we learned a lot. But now we have grown up very much because of the lots of products have come. One suggestion I should say that the regulatory framework has been developed already.
Okay,
I architect the regulatory framework. That's what the country is using to release nanofertilizers. I will share that document.
Yeah, that book
But as a botanist, I would ask something. Because we depend on chitosan a lot. Because for industry like Coromandel, IFCO, all the fertilizer industry depend only on chitosan.
Okay, sir.
Chitosan being a natural source, there is lots of variability.
Oh.
And how to narrow down the variability?
Sir, there are two types of chitosans are there. High,
Okay.
the low. There are three types: high, medium, low.
Okay.
Based on the deacylation process, we have to select.
Right.
So we have to sanitize first.
Yes.
Which one is giving very good results?
Yeah.
That only we have to select. We have to go for the studies.
But information to audience, there is an important source which chitin is rich is a mushroom.
Mushroom,
If someone comes up with pure form of chitosan from mushroom, it is a very great help for industry.
mushroom is very costly, no sir, nowadays. Mushroom is very costly.
No, we are, no, it's, I'm just suggesting, because after mushroom is harvested, the substrate carries chitin,
Mushroom also
can
one
be
of the
good source. You also indicated.
Yeah.
Thank you, ma'am.
Okay, sir.
Thank you for this talk that is really comprehensive. I was wondering, can you combine the nanoparticles that for chemicals with nanosensors to target specific pathogen? Do you think it's something feasible?
Yeah, possible, sir, possible.
You can answer. You want to supplement?
Yeah, please add, sir. Please add. Okay.
See, the nanoparticle acids, there is no specificity. The nanoparticle acid is no specificity. It has to be conjugated with DNA, conjugated with a specific molecule, which sends the thing properly. So we need to dope it. There is a mechanism called doping. Nanoparticle acid is inert. It is only a surface area, we target a nanoparticle. Once it is doped, it can deliver. Even Bharadayar University has come up with a specific drug designed for cancer. It can release only the cancer cell because it is pH dependent release. So nanoparticle serves as a carrier. It carries the molecule, deliver where it is required. Thank you.
So actually I have a short question as well. So you say that commercial applications are limited, but do you have some example for the commercial application in India?
Applications limited, but SAR is told some what is it? Regulatory frameworks are there. If the nanoparticles coming under this regulatory framework means we can apply for we can go for applications. Otherwise, we have to rework and see. We have to see the environmental impact also. This is because finally we will affect the environment only. So we should take care of all the things.
Thank you very much.
Okay.
So now I would like to welcome Dr. Sumiya Sharma from ICAR Institute in New Delhi, and she's going to address predicting plant pathogen interaction using deep learning. Thank you.
So a very good afternoon to one and all. I'm working as a bioinformatics scientist at Indian Agriculture Research Statistics, and this is a topic that I have very recently I've started working on. So this is a very small work that I have recently done. I'll be presenting. So as I work as a Thank you. So I'd just like to brief about the bioinformatics. So it is basically an integrative approach that integrates statistics and computer applications for analysis of biological data. And it involves innovations in computational technologies to accelerate the research in biology, and specifically we do for crop improvement. So from the last few days, we are talking about the plant pathogens, the losses, and the economic damage that are done by them. So we are well aware of the fact that they are a huge challenge for us. So in this scenario, the plant pathogen interactions is very crucial for improving crop protection and productivity. And the resistance or susceptibility of plant is basically defined by the molecular exchanges between the plant proteins and the pathogen proteins. Though we have various experimental techniques that can be used for detection of plant pathogen interaction, but they are costly, time consuming, and less scalable. So that gives us the alternative to use machine learning and computational methods that can be used for prediction of plant pathogen interaction. Various sorts of omics data that we receive for analysis involves genomics data, transcriptomics, proteomics, epigenomics, metabolomics, and phenomics data. And especially after the advent of this next generation sequencing and completion of human genome project, we have huge amount of omics data in our hand that requires advanced computational techniques for analysis. So machine learning gives us an alternative to that. So we are using machine learning to facilitate large-scale plant pathogen interaction identification, because it has already been used for various biological data analysis involving bioimage processing, gene function prediction, protein structure prediction. So that can be used for PPI identification as well. So these terms we come along very frequently, AI, ML, and DL. So briefly, AI is a larger domain that encapsulates the machine learning. That AI is basically making machines mimic human behavior, having human intelligence. Machine learning basically identifies patterns from the data to make predictions. Deep learning is further a subdomain that involves neural networks for prediction. So the basic steps that are involved in the development of machine learning models starts from data collection, then data processing. Feature extraction is an important step because the data we have is in the form of sequence, and first we have to convert the sequence data into numerical format. Though in deep learning, we can directly feed the data and it itself extracts the feature. And then we implement the machine learning algorithms. So based on our problem, whether it is a classification problem or a regression problem. Classification problems is when we have a categorical output. The Y is categorical. Then there can be classes, whether presence of disease or absence of disease, if the proteins are interacting or not interacting. So this is a categorical data. The output is categorical. Then we'll go for classification. If it is a continuous variable, so we'll go for regression problem. That is a regression problem. So in case of quantitative traits, and classification is the case of qualitative traits. If we don't have labeled data, if we just have data for which we do not know what to predict, in that case, we go for clustering techniques, in which we cluster together the similar looking data to get some inference out of it. So there are various evaluation measures that are implemented based on whether it is a classification problem or it is a regression problem. If we use accuracy, precision, recall, F1 score, so these are the measures that we use for classification problems. And mean absolute error, mean standard error, RMSE, R square, these are used if there is a regression problem, and these are some of the indices that we used if it is a clustering problem. So when we have the data, first thing is feature extraction. So what I'll be explaining is basically based on the protein sequence, amino acid sequence data. So the features that we can extract from the amino acid sequence data is amino acid composition, Kremer frequencies. This is basically to convert the sequence data to the numerical data. Then we can extract various physicochemical properties, hydrophobicity, polarity, charge, molecular weight, etc. There are various structural and evolutionary features that can be extracted from the data. Further, if we are not going for feature extractions, we can encode the sequence data into numerical format. So various encodings are available. Most popular ones are the one-hot encoding, bot2vex. So these are the things with which we can convert sequence data to numerical format. So for protein-protein interaction, we can extract basically the physicochemical features, evolutionary features, secondary structures, and these features can be further fed to the machine learning model to train the model and give prediction for the unknown data. So for our study, we have taken the plant pathogen interaction data from these three databases, 5base, HPIDB, and Pathoplan. So they have various protein-protein interaction data. So we have taken basically the plant pathogen interaction data. And further, we have extracted those sequences from various sequence databases, and we have used sequence-based encoding schemes to convert the sequence data to numerical format. For construction of base model, first we have divided the data to the training data and the testing data. The testing data we'll use to evaluate the accuracy, precision, and all those things of the build model. So first we have taken 90% of the data for the training purpose and 10% of the data for testing purpose. Further for training, we have divided the data into 5 subsets. So this is done for 5-fold cross-validation, so that we utilize whole data for the analysis and testing as well. So in 5-fold cross-validation, what we do is take 4 parts for training and 1 part for testing. So we do it 5 times, so that each of the subset is utilized for training once. We basically model three of the algorithm based on 3 of the algorithms, popular machine learning algorithm, that is support vector machine, random forest, and ANN. So best performing models that we get out of those models is and we tested on various descriptors, the feature sets that we have extracted. So based on SVM on autocorrelation, random forest on local descriptors, and SVM on conjoint triads. So these are the descriptors. We have got the maximum accuracy, so we have selected these models. Further, to develop the ensemble model, we have given weightage to these models and the probabilities which we get after the prediction, so to get the final ensemble model. So this is basically the whole outline of the work that we have done. We have taken the data from 3 databases, then we have extracted the features, ran three of the models, and then we have done the ensemble method using the soft voting approach. And then we have got the final ensemble model, and that we tested on other test data that we kept separately before training the data. So after developing the ensemble model, it has achieved the 97% accuracy. That was a bit higher than the individual models. So it shows that the ensemble method is better than the individual approaches. This is one more small computational work that we have done to show how plant pathogen interaction happens. So this does not involve machine learning, but how computationally we look at the plant pathogen interactions. In this, we have taken the polygalactonases. So these are the enzymes that are involved in breakdown of cell wall when the infection happens. And the counter protein that is in plant is polygalactonase inhibiting protein that protects the cell wall. It basically starts the immune response and triggers the defense signaling. So to see if there is a mutation in the PGIP, how it affects its interaction with the PG protein. So this is, we have taken the sequence from radish PGIP and its wilt causing fungi. So for that we have built the protein structure model, validated that through already available information in other crops. We have identified the active regions that are actively involved in the interaction. So we have mutated those regions, and then we further dogged both of the structures before mutation and after mutation. And through MD simulations, we analyzed if the interaction is increased or decreased. So we have seen that after mutation, the interaction between plant and pathogen protein is increased. So it can better help to plant resistance and triggering the defense response in plants. So that could be taken further for wet lab experimentation. In brief, this is the overall AI and plant defense that can be implemented in various defense research. For resistance gene prediction, we can use it to predict gene expression levels in resistance signaling, predicting the optimal levels of defense-related metabolites or proteins, predict stress-related genes or proteins, identify potential biomarkers, and predict the efficacy of various different compounds, the secondary metabolites. So the key takeaways from this is we can combine machine learning and omics for studying the plant pathogen interaction, and it can provide a very cost-effective, highly scalable technique for proteome-wide PPI identification. So I would like to thank fellow scientists, my department, ICR, ISRI, and DBT for the funding. Thank you. And thanks to the organizers for inviting me to this talk.
Thank you for your very inspiring presentation. I'm sure there will be many questions.
Thank you very much for your very pedagogical presentation. It was a great job, so thank you for that. I had a question. You presented a technique to assess the quality of prediction based on the cross-validation approach.
Yes.
And I'm wondering if using this cross-validation method, you may not underestimate the error of prediction of your model because simply of correlation within your data set. So I think it's okay if you manage any correlation within your data set, otherwise there might be a risk to underestimate the prediction error. How do
Actually, as per I understand, cross-validation technique is basically used to avoid overfitting. In the data, so that we use whole data and test over the whole data, so that it increases the robustness of the data.
Yes, but it works only if there is no correlation among the data, because otherwise you will not respect the fact to use some data to design your model and the other to assess its quality of prediction. In other fields, we get rid of any temporal or spatial correlation in order to make sure to avoid this bias in the technique. So it's really a naive question because I am not familiar with omics approaches. So maybe it's not a relevant question. But I would just recommend to make sure that there is no correlation within your data set before using the cross-validation technique.
Yeah, thank you for your...
It's an excellent presentation. You presented very nicely. You have gone through bioinformatic tool to how to predict and all. And is there any success story from Indian Statistical Research Institute with very challenging disease with the bioinformatic tool? You crack down, this is a region you need to target, something like that you figured out?
I personally haven't done any.
No, your institute. For example, this morning there was a presentation on gingerly phyllodes.
Yeah.
We don't have a resistant source. Everywhere it is susceptible. And there are lots of things have been shown. I think genome of sesame is already known.
Available, yeah.
So in that case, you will be able to identify which region we need to target. Can you go with a coal plot or you showed Ramachandra plot and all?
So for that we need the resequencing data for the GWAS analysis and
GWAS analysis, okay.
Then we can go for identification of those SNPs and the QTL regions that are actually involved in that.
Right. So you will be able to figure it.
Yes, if I am having the sufficient resequencing data of sufficient lines that are having
certainly
resistance.
they may have it, no?
Definitely we can do that.
No, you have to say it. Huh?
Yeah. Yeah, sir.
So that if it doesn't have all the genotype,
at least a few pathologists could train.
Yeah, that is the problem, sir. As a pathologist, we are generating a lot of data, sequence data and all. But analysis, especially the bioinformatic analysis, there we are lacking.
Yeah.
Even in TNAU, we are generating data, a lot of data. We have department also, but there is no coordination. That is the problem.
Yeah.
This morning I showed how acid-acting is delivered at the point of interest, how it is dissipated. Everything is resolved with this, molecular modeling.
Yes, sir.
Simulation models. You also run simulation models?
Yes, sir.
How many seconds you ran?
The molecular dynamic simulation for 200 nanoseconds.
Nanoseconds.
Yeah, that is computer intensive, highly computer intensive.
Please. Thank you.
Thank you.
Okay.
Thank you, madam. Thank you very much for your nice person.
A short question, maybe? Just I would like to know if in your approaches you used ordinal variables in the omics data sets, or are they all quantitative or nominal? Do you work with ordinal variables?
No, this is a whole numerical data, not a quantity. The Y is categorical data. This is whether there is interaction present or not.
Okay.
But the X, the explanatory variables, all are the numerical data. It's not categorical data.
Okay.
Thank you.
Yeah, thank you so much, madam. Thank you. So next, I now request our king, the organizer, as our. So Sindhil Raja will be talking about phytoplasma, a growing threat to global food security. Sindhil Raja, please.
Okay, good evening everyone. But it is something different, don't worry. So yeah, good evening everyone. So by looking at this slide itself, you might be having something in your mind. So it's my duty to declare that. No, I don't belong to any particular type of. No. I don't belong to any particular type of plant pathologist or group of plant pathologists. I do work on various crops, various organisms, and I try to integrate the advanced tools and techniques in pathology, microbiology, or nanotechnology, biotechnology, or even bioinformatics or statistics. That's what I do. So, but mainly I have been working with Pyricularia, Phytophthora, and Phytoplasma. These are the main three phytopathogens I am working for the last 10 years. Pyricularia, anyway, that is a serial killer. Phytophthora, that is a plant destroyer. Maybe you can call it the Phytoplasma as a. Yes. There was a huge outbreak in Gudalur. And maybe Phytoplasma, you can call it as a silent killer, I could say. So, but today it's all about the Phytoplasma. Why we have chosen this topic? This is the work of 4 students who are sitting at the back. Okay, I'm summarizing because we have chosen this topic. All of a sudden, there were a huge outbreak of this phytoplasma everywhere. You could mainly previously it used to be a problem of sesame, then brinjal. Later it moved to coconut. Now, if you go to the field, everywhere you could see this phytoplasma incident, but not at the alarming level. The level of intensity or incidence may be minor, but it is occurring everywhere in all crops, irrespective of agriculture crop or horticulture crop or weeds, aromatic plants. Everywhere you could find the phytoplasma. But with regard to other pathogens, there have been many work done by various scientists, but in case of phytoplasma, there is not much work done on it. So that is the reason we wanted to work on this organism. So we allotted some topic to the 4 different students to develop a holistic approach, or how to. First thing is we need to characterize, we need to understand its evolution, because when it comes to phytopathogen or plant pathogen, there is a systematic classification for bacteria, fungi, viruses, but for phytoplasma, it is evolving continuously. There are different groups. So far, 34 groups have been reported with more than 100 subgroups. But it is keep on continuously evolving. So that is the reason we wanted to understand the mechanism of this organism, how it is causing disease to the various crop plants. Okay. So in the case of phytoplasma, mainly it is using various mechanisms to cause different symptoms. Mostly it is symptom may mimic like viruses. So as far as you said, there is no any control measure for this, at least for Sesamum pylori, this phytoplasma disease. You cannot use resistant source, or even you cannot apply chemicals to control the disease. So somehow we want to control this disease. There comes the approach. So first thing is we need to characterize the group, at least in Tamil Nadu. Throughout the world, as I said earlier, there have been 34 groups. So we wanted to know what are the groups present in Tamil Nadu. So that's why we have chosen the topic, and we have allotted one thesis topic to assess the geographic distribution of this phytoplasma disease in various crops in Tamil Nadu. Then we wanted to characterize all the groups, and we wanted to develop on early detection kit. You may say that symptom is visible, why do you want to develop the kit? But at least if you develop the kit for early detection, it may be good for recommending some management measures to contain the level of incidence. In some other cases, cereals, the symptom may not be visible, it's asymptomatic. So in that case, it can be useful for at least scientific point of view to diagnose the disease. So characterization, diagnosis, and finally comes the management. We tried different methods, chemical, physical, and biological, and even resistant gene, the gene bank from the NPPGR. We got lot of resources, around 6,000 accession we screened for, at least for sesamum. None of them are working again, are giving resistance to the disease. So the approach is to develop at least one resistant variety. So here you could see in Tamil Nadu, so it's occurring almost 16 different. So far, whatever we have documented in major crops like chili, vegetables, apart from the sesamum, other oil seeds like groundnut and vegetables, aromatic crops and coconut also. There are many, including various weeds also getting affected with the phytoplasma. So that we have documented. Next, we have characterized the organisms. So we found that out of 34 groups, 4 groups are present in Tamil Nadu. Yeah, only in Tamil Nadu, not in entire India. In Tamil Nadu, we found 4, there is 16 SR 1, 2, 5, and 6 group with 9 subgroups. So that is one thing. And you know the symptom, it can cause different type of symptoms. So the challenges in working with the phytoplasma, is non-culturable one. We cannot, that is the difficult. We made some attempt, but we couldn't able to culture this organism. It is unculturable one. And because of this obligated nature, it will colonize the flowing region. Still, it is very complex to isolate and characterize the organ. That is one thing. Another thing is it is maintaining the tri-traffic interaction. Like a virus, you have to study about the host, insect vector, and the pathogen. When it comes to vector, again it is a complex. Previously, people used to say there is only one, mostly like the hemipteran pest, leafhopper or plant hoppers are the vector of this phytoplasma. Previously, people used to say there is only one vector responsible for the particular phytoplasma disease. But that is not the case nowadays. If you go to the field, you could find a complex of phytoplasma. So it is a mixture of phytoplasma, not a single phytoplasma causing this same disease. Various phytoplasma spread in the same disease in the field. Say for example, whether it's a brinjal or sesamum or whatever it is. So that is another thing. And there is not much molecular work to dive into this phytoplasma to study its characters. or the interaction between the host pathogen. So these are all the various challenges working with the phytoplasma. Still it is keep on continuing. There are stalwarts working on the phytoplasma, but still it is very challenging too. So if we want to understand the pathogen and to control, we should link the or study the genomic data and the practical challenges in detecting this pathogen well in advance, the field condition. So for that, so for that, we decided to develop one RP assay. Mainly we thought of developing some LF based kit, but if we end up with RP assay for there are advanced PCR methods are there, but they are time consuming. But with this method, we can diagnose the disease within 30 minutes. We don't have to wait for 3 or 4 hours like PCR. That too with a crude extract. You don't have to extract DNA also. So here you could see the sensitivity of this method when compared to PCR. Even if you extract DNA, you can also use the DNA for this RP assay like PCR. The results will be same. But when you extract the crude and directly if you use for PCR, it won't work. But for RP assay, you can get the result within 30 minutes. It's just a thermal amplification method. You don't need sophisticated instrument or laborious or time. There is no such a case. Okay, within 30 minutes, you can detect this disease by using this method. So this is one approach. So next one is the management aspect. I think something is missing here. So probably you might be knowing about the gene for gene hypothesis. Right? No. So in the biological system, everything is governed by gene, right? So here we wanted to study, if you want to study the interaction between the pathogen and host, there must be some resistant gene or susceptible gene in the plant system, and correspondingly there will be some virulent factor or virulent gene or pathogen factor in the pathogen. So we want to crack down that one. But here in case of phytoplasma, the SAP54 effector is the main culprit causing the disease in various crop plants. That is one thing. Correspondingly, if you see the plant system, I will come to the later, because if you see the symptom of this phytoplasma, it will look very unique. It will convert the floral system into leaf-like. It will modify the floral parts into leaf-like system, or you may find some malformations. It's all because of this. It will go and directly interact with the mat-spark system, which is highly essential for the development of the reproductive structure in the plant. That's why all the time when the plants are getting infected with the phytoplasma, it will become sterile. They will directly go and infect the mat-spark system in the plant system, so they cannot be able to produce flowers or fruits or whatever the grain. So but these genes are very essential. They are all the susceptible gene. But if you go and touch or edit the mat-spark system, the entire plant system will get collapsed. Again, people started working on it. Finally, they found that there is a shuttle protein between the mat-spark gene and the effector protein in the pathogen. There is a protein called RAT23, which is acting as a shuttle protein between this pathogen protein and the plant protein in the plant system. So they have decided to silence that particular gene, so that the pathogen protein cannot recognized by the plant system for a compatible interaction. That's why I was talking about gene for gene hypothesis. When there is a compatible system only, the plant will produce disease or symptoms. If it is incompatible, it will become resistant. So the thing is here, the RAT23 protein in the plant is the susceptible factor responsible for causing the disease, phytoplasma disease in the plant system, whereas the effector is a virulent factor in the pathogen. So we cannot do anything with the phytoplasma effector protein, but at least we can restrict the we can knock down the susceptible factor in the plant system. So with that approach, we identified and characterized the SAP54 effector protein in the phytoplasma in all the groups, and simultaneously here we took the sesamum as a model plant here. So we have quantified the effector expression in the infected plant. The level of expression is varying after infection from 15 days to till maturity in all the plant parts like stem, leaves and flowers also. It is having the expression, the phytoplasma expression is there. So as I said earlier, there is no any method to control this disease, particular disease. So with the only approach, genome editing only, we could able to develop the resistance in the plant system. So we have identified the target gene as or susceptible factor as a RAT23. So here we have developed the guide for guide RNA for this particular protein, and with the help of genome editing, we could be able to transfer the guide RNA into the plant system. So that will give resistance when there is no susceptible factor or particular corresponding gene in the plant system. The pathogen cannot be able to recognize, and it will cause the disease. So that is the idea. So with that idea, we have the work is half phase still. We could be able to develop the tissue culture plant, and we identified the target gene, and we have developed we have transformed the gene. So it is still under evaluation. We need to go for the phenotypic assay and through transmission insect assay, as well as we also developed the clone for that. SAP54 effector clone is there. With that also, through agro-infiltration method, we can prove the pathogenicity. Either through agro-infiltration method or through the conventional insect transmission assay, we are trying to evaluate its efficacy or its resistance under field conditions. So that work still is going on. So this is the only way to work with phytoplasma. First thing is we need to characterize it, and we should develop some early detection kit, and we should study and characterize the effector protein present in the pathogen system, because that is the main culprit or the factor responsible for causing the disease in the plant system. And the only approach I could feel the genome editing. Yeah, genome editing would be the option for developing the resistance. So that is missing, and time. Okay. Thank you anyway. That is the thing I want to convey here. My student. Yeah.
So, thank you so much, Dr. Sendil Raja, for your nice presentation. And, so I have one basic, curious to know why nowadays this phytoplasma disease is becoming serious, whether it is due to alterations in the genomes of the phytoplasma or through the vector diversity.
There are many factors involving here. With climate change and new pathogen, many things are happening with co-evolution and all. We, as morning session, Professor Kumar was telling about we need to model the effector and all. We also tried that approach, because when I was talking about the effector protein, the same effector protein is causing, you might be knowing that the same phytoplasma species is causing disease in all the crop. As a pathologist, we know that there are group host-specific and host-non-specific organisms. Certain pathogen only cause disease in particular disease crop. Certain, like Polyphacus insect, it can affect a wide range of crop plants. Similarly, in case of phytoplasma, if you see the scientific name, Candidatus phytoplasma, it will come with some species name, Aurantifolia or something else. But the same species causing disease in all range of crops, wide range of crops. So we wanted to know that what could be the reason. For that, we have tried to model the effector protein, how it is evolving over a period of time from one place to another place or from one plant to another place. But we made an attempt, small attempt through some computational approach. We found that the adaptive nature of that effector in all crops, whether it is a resistant plant or susceptible plant, or whether it is a vegetable crop or monoecious crop, whatever it is having the adaptive nature to cause but still we need to work on it. It is having adaptive potential to survive in all type of plants.
Yeah, majority of the weeds are
weeds, horticulture crop, same, the thing is the same species. It's not like a pyroclera is having different strains, other pathogens are having different pharma species, but in the case of phytoplasma, the same species causing disease in different crops. That still we need to work on. That is
and similarly, even tetracycline become ineffective.
Yeah,
those days there are reports tetracycline found to be effective at least for 1 or 2 months. Remission of symptoms will be there. Now that kind of reaction is also not there. So that's why I wonder whether major changes in the genome.
Yes, evolution and genoplasticity.
You already identified which region you are going to target. Which region?
RAT23 is the protein. Yeah.
And you identified the system what you are going to use.
No, we are going to remove that. Yeah, that's it. That is the approach of genome editing.
Now, what about madpox?
No, we are not going to touch the madpox. We are also trying that one. If we touch that one, maybe the plant system will get affected. So instead of madpox, the RAT23 is acting as a shuttle protein between the madpox and the pathogen protein. So it is better to target the RAT23 instead of the madpox, because it is highly essential for the floral and other reproductive organ development. So we don't want to touch that upon. Not only in sesamum, in all the crop plants, the mat spark is the reason for the development of flowers and.
Use whatever the tools previous speaker explained.
Okay, yeah.
Then narrow down. It was a very good one. Okay, you are telling that normal picture you are able to identify the phytoplasma in gingerly, is it right?
No, through conventional PCR also we could able to identify in all phytoplasma. But if you want to time consuming, it is.
Okay, the thing is in citrus greening, we are unable to detect the phytoplasma with the normal crude extract. So what we did, we remove the veins, then veins only we did the PCR, then only we got the bands. So have you tried such kind of any, after dissecting the vein, because wherever the phloem is there, that is persist. Have you?
Citrus screening is different organism.
Okay,
not
but the phytoplasma
but
present
the
in the
thing is,
phloem.
if you want to develop a
That's why
user-friendly kit, if you want to develop a user-friendly kit based on the LFA method, it should be very user-friendly. You have to extract the sap extract and, from the plant or leaf, whatever it is. If you add your reagent, it should work immediately. Otherwise, it's of no use.
No.
There should not be any comparison, tedious process.
Gingerly is okay. But in
No, but for other
no,
process also, other plants also, we should develop such reagents to quickly degrade the plant parts and to get the extract or sap from the plant. Otherwise, there is no use of developing such a kind of kit. No, all nested PCR will work with all plants. No, citrus greening is different
it is
organism.
mainly due to uneven distribution only.
That is there.
Sometimes wherever no, we take this tissue that may not contain the phytoplasma.
Yeah, I have. Thank you for this very nice introduction for me, who doesn't know anything about phytoplasma. But I had a question for you, Dr. Santil King. I was wondering, from a practical point of view, is it efficient in terms of economic return to perform the loop that you showed on the diagnosis and all the molecular techniques, to then deploy some management technique in the field? Did you conduct some economic analysis of the system?
No,
so that it could be spread at a large scale for farmers?
Yeah, the study is still halfway, it's not yet over. And another thing is, once if you develop a resistant variety, we could use the seeds for further generation. So there is no need to develop tissue culture plant again and again. We can use the seeds, so there won't be much cost for that.
The management is focusing on a vector, right?
Not.
That is what he is asking. What is the management strategy
No,
you can
he is
follow?
asking about the economics of all these studies.
Yeah, economic benefit, it could be a resistant type or it could be a management strategy.
But once if we develop the resistant variety through genome editing, we are not going to develop the tissue culture plant again and again. We are going to use only the seed material.
But resistant variety, lots of complications were presented this morning.
Yeah, but government of India also ready to give the consent for using. No, there is no resistant sources.
Then how are you going to do that? That is what I asked.
No, that's what I am telling. Through genome editing only we are going to develop. Not through other methods.
So you must get a project in genome editing.
I already got one. That is working.
Is confirmed.
Yeah, thank you. Anything else to do? Thank you. Thank you.
Thank you. And we will have the last presentation of our seminar that will be given by Dr. Angapan from Tamil Nadu Agricultural University of Coimbatore on the impact of antimicrobial resistance in agriculture, a One Health perspective. Thank you.
Thank you. So I am going to talk about the impact of antimicrobial resistance in agriculture. So we are in a very basic state of research now, very infancy. So I am going to give an overview. It is not from our research finding. So I am going to give an overview of what is happening around the globe on the impact of agriculture on antimicrobial resistance. So you may aware of the antimicrobial resistance. Long time back, decades back, we used to think about that antimicrobial resistance means antibiotic resistance. So now it is not like that. It encompasses antifungal resistance and other resistance which are developed in the microbial species. So during the month of August, you might have aware that there was a news clipping. There is a pathogen called Candida tropicalis which infects a human being, which is an invasive organism in tropical countries, where it causes candidiasis, a disease called candidiasis in human being, especially in bloodstream. So now people are struggling to control this particular disease, because for the management of that particular disease in human being, azole group of compounds are used, a chemical called fluconazole, which was given for the management of that particular disease in human being. Now, even after the increased or elevated dose of this particular fluconazole, they could not able to contain the disease. When they examined why, what kind of reason, why it has happened, so they found that that has developed resistance due to the use of a fungicide called tebuconazole. So tebuconazole is now commonly used by the farmers for the management of several diseases. So the organism has developed cross resistance. So it is very difficult because no one has imagined that it is mainly due to the use of a fungicide which is for the management of plant diseases could able to cause a resistance in human pathogen. So likewise, in previously a decade back, we used to tell the restricted use of antibiotic compounds. So everything is in theory, not in practical. So there is no regulation for the restricted use of these antibiotics as far as India is concerned. We are having a lot of antibiotics which are used for the management of plant diseases, which is available. So just we will see this, what is this antimicrobial resistance. Just last year only we have initiated, for one of the master students, we have given a topic on the phytoplasma resistance. So next. So the antimicrobial resistance is the ability of the microbes to resist antimicrobial agents. It is a broader term. So it is a global challenge, and also it is linking to plant, animal health and human health. It is not only concerned with human health, it is also concerned with animal health and also the plant health. So that's why it is nowadays considered under the One Health approach. Very recently, last month, we got a communication from our state planning commission. What kind of research work you have initiated for this One Health for the management of antimicrobial resistance? So we have given a reply that we have initiated. It is in the very primitive stage. So in agriculture, the extensive use of antimicrobials for managing the crop and livestock diseases. So agriculture includes animal husbandry, fisheries, shrimp farming, several things. So most of things the people are in animal husbandry also people are using antibiotic compounds. In fisheries, in shrimp farming, they are using lot amount of antibiotic compounds. Livestock diseases that leads to the emergence of resistant pathogens and the spread of the resistant genes. So two things. So here it is the AMR. One is development of resistant pathogens and also the spread of the resistant genes from this organism to another organism, maybe of the same species or a different species. So this led to the persistent infection. So as I said earlier, in Candida tropicalis, so it leads to the persistence of the infection in the human being or animal or in the plant system, and also what it leads to? It leads to over usage of the chemical. So that leads to environmental contamination. So normally in farmers' practice, if you recommend 2 ml per liter of water, they used to go for elevated dose for the management of either pest or disease, whatever the case may be. So if it is not controlled by the particular chemical, they are tempted to use more quantity. That leads to a lot of contamination in the environmental system. And also it affects the ecosystem balance. So we are talking about the soil health. Soil health depends upon the soil microbiome. So such kind of resistant microbes are present in the system. They will affect the balance of the microbial community in the soil. That in turn affects the soil fertility, soil nutrition management, conversion of nutrients, several things. It is a chain of events. And also ultimately it leads to the food safety. So Dr. Sar has told in the morning that you can test your the presence of pesticide residue on a peeling method, just touch and peel. So like that, here it leads to a lot of contamination in the food products. So that's why it is a main culprit for several aspects. It is not only directly antimicrobial resistant, and also several diseases in human being, especially one of the presenter, I think on the first day, they told that in a particular village there were so many number of cancer patients because of use of large quantity of this chemicals, pesticides or fungicides, mainly due to they are dumping, mainly due to they want to control and harvest. But it is not on the dose which is recommended by the scientist or by the extension worker. Overdose is also leads to the development of resistance in the organism, so that ultimately leads to the affect all the food chains in the system. So the One Health concept, it is the approach, it concern about the health of humans, animals and environment. It is an interdependent word, One Health. And this antimicrobial management integrates surveillance and interventions and across the system, because if we are alone, not alone, plant disease management or insect management, not alone going to solve this issue. It requires the intervention from all the sectors and also surveillance from all the sector. Then only we can able to have a fruitful result in this particular management. In agriculture, this AMR highlights the resistant bacteria and genes move through soil, water and food chains. So that's what I explained, that it is moving through different systems, directly and also indirectly. So this is the discovery of antibiotics. So right from the penicillin discovery, in the above, it is mentioned the year of discovery of the antibiotic compounds. In the below, you can say that the arrow marks indicates the development of resistance against the particular antibiotic compound. So most of the antibiotic compounds which were developed up to 2000 has developed resistance. So in Indian scenario, as I said, that the research in this particular area is in a very infant stage. So we have to generate lot of data, and also first of all it requires the collaboration and coordination between different departments like human welfare, that is medical department, animal husbandry department, fisheries department and also agriculture department. So there are several departments which are operating or working in isolation, but it requires an interaction and coordination among the different sectors. So that's what I told our state planning commission. They have called the veterinary university people, fisheries university people, and also from medical science. The member is retired medical professor Dr. Amlor Pavanathan, who only chaired the session. So this is the absence of strict regulations. As I said earlier, we don't have any strict regulation to comply with the restricted use of antibiotic compounds. So that is a major issue in case of, say, I may not recommend the antibiotic compound, for the management of the disease. But the farmer can get it from the shop directly, because there is no restriction for the selling of that particular compound. So it is available. It is available in the market. They can use it. So there need a restriction for the production and the sale of these particular compounds, which are the culprits for the AMR. So nowadays, as I said, it is an infant stage. National initiatives, academic research are gradually bridging the gap. So now only in academic institutes also started working on this line. And these are all the hot spots for the AMR, agriculture, animal husbandry, household, industry, even from the sewage water from household or industry or hospitals or aquaculture, the groundwater contamination. There are several sources which led to the AMR. So as far as we are focusing on plant pathology, in plant pathology also we are using antibiotic compounds for the management of bacterial diseases, streptomycin, tetracycline, kasugamycin. These are all commonly available antibiotics for the management of diseases. As during the previous presentation, Dr. Karthikeyan pointed out that tetracycline was recommended for the management of phytoplasma. It could be able to suppress the development of the symptom for a brief period, and afterwards the symptom remission will be there. But nowadays, even we could not able to see the symptom suppression at all. So that itself indicates that it is the particular organism might have developed resistance against the particular antibiotic compound. So this kind of resistance development hampers the disease management, increases the dependency on stronger, often more hazardous chemicals. As I said, the farmers are forced to use overdose. So that is also harmful to the environment and also the human being, in the food chain, everything. So these are all some examples. Some of the antibiotic compounds which I have mentioned were already reported for resistance. Say example, Xanthomonas voricae, Petovar voricae, which causes bacterial blight in rice, which is showing the antibiotic resistance are reported for streptomycin, tetracycline. In tomato, Pseudomonas syringae against streptomycin, citrus, potato and chili. Some of this is a very primitive work they have reported from the, I have culled out from the literature. So what is the mechanism by which this AMR is developing? So enzymatic inactivation in the system. So these are all the mechanism by which it leads to the AMR development. And in case of the acquisition of resistance, normal bacterial things, transreduction, transformation, conjugation, all those things are aiding for the development of AMR. So these antimicrobial genes are normally moved through the conjugation process from the resistant bacterial cell to the susceptible one. And horizontal gene transfer also abating this kind of phenomenon in bacteria. So this impact is that they are affecting, it reduces the soil fertility, nutrient cycling, plant resilience, and enhancing the pathogen survival. And what are all the, there is a national action plan for this AMR. The objectives are rational antimicrobial use, AMR surveillance, promotion of alternatives, and capacity building. So these are all the some objectives of under NAP. So there are throughout the nation we are having a network of surveillance laboratories. One is situated in Coimbatore also, Coimbatore Medical College. We are having one surveillance laboratory. So throughout India we are having 30 different laboratories. So the molecular surveillance approach is one of the tools which we can employ for the identification of this antimicrobial resistance. And also now we are having a basket full of molecular tools like metagenomics, which are helpful to identify these bacteria which are present in the soil. So the rhizosporic soil or the field soils we can subject to metagenomic analysis, and we can identify the presence or absence of this antimicrobial resistant bacteria. And resistant gene profiling is another method by which we can identify the bacteria which are possessing the resistance against a particular group of chemical or compound. So now we are in the era of artificial intelligence, so we can also employ artificial intelligence for the AMR prediction. This will help us to integrate the AI with genomic data, which accelerates early detection and decision making for the AMR management. So sustainable alternatives, what are all the sustainable alternatives? We can, very popular is biological control. So avoiding the use of the highly toxic chemicals, we can go for biological control, and other bioagents like bacteriophages and antimicrobial peptides can be used for the management of this AMR, to suppress or minimize the development of AMR. It is not going to totally wipe out, we can minimize. And genome editing strategies can also be employed. And ecological monitoring and mitigation, this is a very, very important one. So, because I told, I have given in one slide I have showed that the sewage water contamination through the industry or hospitals, several things we have to monitor. So the ecological monitoring is very much important for this AMR. And also we have to create awareness among the farmers. That is the first point we have to do. We have to tell what are all the harmful effects of these chemicals and the AMR, and how which can be minimized or mitigated. We have to create awareness among the farmers, and also we have to tell them the alternative strategies by which we can able to overcome or subsidize this kind of development. So there are global and regional collaborations are there, FAO, WHO, World Organization for Animal Health, World Global Antimicrobial Resistance Organization, GLAS. So there are several organizations are working. So they are developing databases. So these databases can be enhanced by cross-border collaboration, because we need data from not only from one country. Throughout the world, we need data on such kind of activities. Then only we can able to have a concrete idea how to manage this. So there is there are a lot of challenges and knowledge gaps available. So for this management of this AMR. So the future prospects is we can expand the AI you can use the AI analytics, expanding the AMR database, and promoting sustainable agricultural practices, as I discussed earlier. Strengthening of interdisciplinary research, that will be the that will give the holistic approach for this tackling the AMR. So, nutshell, AMR in agriculture threatens food security and also ecosystem stability. So, One Health approach, integrating environmental, plant, human health perspectives is essential to mitigate the risks. The future lies on sustainable innovation, policy reform, and global cooperation. So, it is a global problem, not only a problem pertaining to India or France. So, we have to collaborate with world level organizations. So, it is a global issue we have to address to mitigate this kind of AMR. Thank you very much.
Thank you so much, Dr. Rangapan, for nice presentation on the antimicrobial resistant development and their mitigation. So now the paper is open for discussion.
Thank you, sir, opening eyes for the new area of research. This is also bring as a challenge or question ourselves to, it's just kind of, human is a multi-drug resistance.
Yeah.
Okay. And this is also if we are spraying fungicide with a pesticide and farmers are being practiced. And this is have you come across any evidence in India in any of the crops, either in cereal or in legumes, mostly the field crops, as such AMR has been reported and it is very serious to look after. So the group can also discuss in those areas, okay, this is the priority crop or disease should think upon. So that takes, it's not just pathologist, and should also bring the molecular biologist and how things can be taken forward. Just your experience and research experiment, do you have any evidence? Thank you.
So I have mentioned a few crops like tomato. There are some isolated incidences. It is not in a large extent. Nice, say example in case of tomato, they have reported from Karnataka, because we don't know whether the entire Karnataka. The paper mentioned about the tomato which is infected by Pseudomonas has developed resistant against streptomycin. The state is Karnataka. So because we don't have an extensive research on this particular area, especially in India. We don't have. So even during our teaching in the classes also, we used to tell the students not to recommend the antibiotic compounds for the management of bacterial diseases. Instead of that, you recommend the copper-based fungicides for the management of bacterial diseases during the initial stage. These are all our own knowledge that we are disseminating, but there is no record that this is there in Tamil Nadu, in Delta it is there, or in Thiruvallur district it is there. There is no evidence-based records are not available. So that's what the State Planning Commission has insisted upon, to record all those incidences and what are all the mechanisms which we can suggest for the management of this AMR. Now only as far as in our country, there is a national action plan for this particular AMR management. Though it has initiated in case of human medicine long time back for the multi-drug resistance against tuberculosis. So now only we have our eyes are open now only. So we are because it is now only people are thinking that it is now in isolation they cannot do anything. So it requires a collaboration between animal husbandry, fisheries, human medicine and agriculture. In addition to that, we have to create awareness among the farmers. We have to develop some sustainable management strategies. So these are all now only gaining momentum. So it is not only pertaining to India. Most of the countries are we are sailing in the same boat. They are in most of the countries they are working in isolation.
Yeah, thank you, Professor. I wanted to share one idea and ask you one question. So you mentioned that you had a national action plan in order to limit the resistance to antimicrobial. And in France, actually, we also had two versions of a national action plan in order to limit the use of antimicrobial for animal health. And actually, this plan, on the contrary of the National Action Plan for Pesticide Resistance, where the results are so-so, this plan worked in France. So we had good success with that. And I was wondering, in the strategy, in order to limit the creation of resistance in the pathogen population, if you considered an integrated approach based on two pillars. The first one is to combine other means of diminishing the size of the population, so using other like plant resistance, cropping practices adapted, or physical or biological control. So that would be the integrated approach to limit the size of the population, because if the resistance is a consequence of a mutation, then you will mathematically reduce the probability of appearance of a resistance. And the second pillar will be a reduction in the selection pressure, either by combining a different family of antibiotics, either in space or in time or in both. So it's just an idea. Do you have some thinking about this more general approach to limit the appearance of resistance, using what can be done for resistance against fungicide, insecticides, or herbicides? Okay.
So, as you suggested, without the knowledge on this antimicrobial resistance, nowadays, say, example, the companies they are producing. So, without the knowledge on this AMR, so nowadays the companies are producing lot of fungicides in combination. Combination fungicides, even companies are producing fungicide plus an antibiotic compound. So because naturally their dosage will be on the lower side. Say example, I am recommending 500 ppm of streptomycin sulfate. But in a combination, we used to recommend it won't come to the level of 500 ppm. It may be a sublethal dose. So that kind of things are leading to the development of resistance, antibiotic, antimicrobial resistance. So in our university, we had a policy. We had a policy not to recommend any combination product in case of fungicides and also insecticides for the management of pests and diseases. We had a policy. But we are not the authority to limit the companies. We can do research, we can teach them, we can demonstrate them. But because it is a different sector, which is manufacturing and marketing, that is a different sector. But they are nowadays, if you go to market, you get all combination products, more number of combination products. So that might be one of the reasons for the development of resistant strains even in plant pathogens.
Okay, thank you.
Thank you.
Thank you for this great talk. I had a question about the development of numeric agriculture and the optimization of machinery, for example, for pulverization of chemicals. Is this something that is really developed in India or not? Like, for example, forecast models for the main diseases and the optimization of pulverization systems, for example, and robotics, maybe?
So we are having our own forecasting models are there. I think Dr. You only presented for the potato late blight, he has presented a model. So we are having the models. We can able to demonstrate, but the end user is not ready to take up our recommendation. So here still, that's what I told, we have to create a lot of awareness among the farmers. Here in our country especially, mostly the farmers will first approach the person who is selling the agrochemical dealers. They are their doctor. So they recommend what are all the chemicals they are recommending and the dose they are recommending, they go and spray. So not most of the time, because it is a profit oriented business for the agrochemical dealer. So they used to take chemicals, they go and spray in double the dose or triple the dose. So ultimately that leads to the development of resistance in the pathogen and also environmental contamination. It affects the soil balance of microbes, soil nutrition status, several things. It's a chain of event. But here in the forecasting model or the model developed, it will give the measurements, what are all the management measures which you have to take. But people are not ready to follow it. That is the major problem. We are having for selected crops, we are having the models. For apple scab, for late blight of potato, we are having the models. But people are not ready to follow it. They used to follow it only for to know that whether the disease will come or not. That's all. The other part, the management part, they depend upon mostly on the agrochemical dealers. That is the main reason for dumping lot amount of chemical in the field.
Thank you, sir. Thank you so much for your presentation as well as a long discussion. Thank you.
Thank you very much.
So, there is an announcement from the organizers. We are breaking for tea now, just for 10 minutes. Then we are again gathering for finalizing the proceedings, final panel discussions. Thank you. Thank you very much.
சரி அது வேணா யூனிவர்சிட்டியில் இதுலாம்
அது இல்லாமல் இன்னும் நாலு
சரி வச்சிருக்க பார்க்கலாம் அது தேவைப்படுது ஏன்னா டிஆர் கொண்டு வந்துருக்கோம் எடுத்துக்கிறீங்களா சரி எடுத்து இன்னொரு ஒன்று பரவாயில்ல
ஆமாம்
இல்லை அப்படி இல்லை நீங்கள் வர மாட்டீங்களே நீங்கள் பெருசாக சீட்டில் இல்லை இல்லை இஷ்யூ இல்லைங்க நீங்கள் ஏதோ கவர்மெண்ட் வந்து கேட்குறாங்க அதை உங்களை கட்டி கழிக்கிறாங்க அங்கே எனக்கு வாங்குறேன் இவங்கெல்லாம் சீடுனேன் சரி ஏதாவது சம்திங் பெக்குலியர் ஆமாம் நேற்று இருந்தால் டெல்லியில் இருந்தேன் இல்லைன்னா அதெல்லாம் இன்னைக்கு இன்னைக்குலாம் நான் ஃபெசிலிட்டேட் பண்ணுறேன். இப்போ டேபிள் மாற்றணுமா? அப்போ அப்படியே உட்காந்து அப்படியே உட்காந்து ஓகே ஓகே what DNA thinks that what you want to achieve in DNA என்னெல்லாம் எதிர்பார்க்குறீங்க இந்த டிஸ்கஷனில் எப்படி கொண்டு வரலாம்
all fronts teaching is such teaching non-knowing respect wise we had a discussion with him and I think during we discussed about the ICR 2003 so we want to
நம்மளே ப்ரொனவுன்ஸ் பண்ணி அப்படியே க்ளோஸ் பண்ணிடலாம். ஏன்னா ஒவ்வொன்றா ஓப்பன் பண்ண இது பண்ணிட்டு ஏன்னா சிலதெல்லாம் நடக்கும், சிலதெல்லாம் பணமெல்லாம் எப்படி அக்ராஸ். ஆமாம் ஆமாம். அதனால் வி மேக் ஷுர் தட் வி டூ திஸ். என்னது டூபிளோ அதை பார்ப்போம். அது மாதிரி
ஷார்ட் டேர்ம் பீரியட்
ஏன்னா அவங்களுக்கும் யூரோப்பியன் யூனியனும் பணம்லாம்
மாதிரி தான் இருக்காங்க உலகம் முழு. எவர்கிட்டையும் பணம் கிடையாது. எல்லாம் இது பணம் இல்லாமல் மனம் போடலாம். அது மாதிரி பண்ண முடியாது.
ஏதாவது போய் மற்ற கண்ட்ரில சுரண்டி வச்சுனால என்ஜாய் பண்ணிட்டாங்க அசட்டை. இப்பெல்லாம் தீர அளவு
எல்லா யார்ட்டையும் இல்லை. அதாவது நம்ம இதாக இருக்கணும், மியூச்சுவலி பெனிஃபிஷியலாக.
அதெல்லாம் தெரியல எனக்கு, நான் அதனால தான் நானே வந்து கூப்பிடுறேன்.
So if you don't mind, like, they don't know what is in the gift hamper. I'll just tell you.
Gift?
Hamper.
Explain.
Yes, sir. Respected delegates, I hope everyone have received a gift hamper. Yeah, we have received it from Ooty. It has the dried rosemary leaves, which has a good medicinal value. And this is dried rosemary. Yes. This is the dried rosemary leaves. And this is a premium Nilgiris tea, like local brand, and it's premium. Then this is a snack made out of wheat flour. We call it varki, Ooty varki.
Okay, the another important thing, while you flying, don't take this. Yeah, because don't, it's a very problem. It's not problem, but you have to be answerable for a lot of things. So better you avoid this one. No. Like, or you have to explain, then you have to put it. So tea is okay, The tea is, doesn't mean. Yeah, it's okay.
All of you here, we'll conclude after. We'll also
You have to be careful. Okay? Yeah, sometime in your country they may ask. Okay, here they won't ask.
These substances look very contraband substance.
That's why
they'll marijuana or something like that. You should go to Netherlands and bring it.
It's okay, because this is a branded tea, all knows.
Yes.
But doesn't know.
Yes.
Snacks won't problem. Only this one you have to be careful.
Actually, this is a medicinal plant, but you put it in hot water and put a little bit of sugar and drink. Yeah, and tea, you all aware about tea, which is very famous. Ooty is very famous. We call, Queen of Hills. That is the place the tea is coming from. So that's famous. This is a cookie, like a Tim Hortons or some cookie. This is made of wheat flour, and you can, in the evening hours with the tea, it goes well. Okay, that's all.
Okay. So we come to the last part of program. So as part of the event, we need to have the group discussion and networking. For that, I will ask Professor KSS to moderate this session.
Okay. Good evening to all of you. Thanks to coordinator Senthil Raja as well the other French coordinator. It's a very excellent program. So now we are in the final phase of discussion. We were planning to have a panel discussion, group discussion, and come up with some specific suggestion. Because we are running out of time, we can make it short. We got feedback from individuals from French group, everybody contributed feedback. And we also get feedback from ICR scientists. We have some information from Agri University. Noel, John Noel, we are waiting for you. See, he gave very good information. That would be the base in which we can build on what we do next. So my understanding is that I handle several international projects, like a Canadian project for several years. And we have taken different levels with the Canadian project. For example, because I got a Canadian project, we got an education program, we have a research program, we have an exchange of students, we have several programs branch out. Similarly, this program will lead to like this. So I would say that there are 4 or 5 points I will say, and for each point, if you have any suggestion, you can say it. Overall, based on this exercise we had, discussion we had, we can take it forward to different levels, 5 points for discussion. Number 1 is education, because at Tamil Nadu Agriculture University is an education institute, and we have undergraduate, PG, PhD in different disciplines. Certainly, we can take advantage of Tamil Nadu Agriculture University being the lead state agriculture university in the country, ranking very high. So certainly, we can take forward the education program. In education program, there are 3 things can be done. First would be at the undergraduate level. So we developed undergraduate dual degree in which the students of Tamil Nadu Agriculture University spend some time in the academic institution in France, and so that 2 degrees can be obtained. Whether there is a possibility. So I just say an example for, we have collaboration with the Nova Scotia Agriculture College in Canada. Currently it is Dalhousie University. What we do, the 4-year undergraduate program, the final year, the students spend time in Canada. Then they get 2 degrees. 1 degree from Tamil Nadu Agriculture University as a BSc Agriculture. Another degree they will get in the name of landscaping horticulture, because it's not a heavy loaded course. So this is what's articulated, developed by me in the year 2008. It is working well for 15 years. Whether such provision is possible, I open to our friends in France. Is there any possibility? Can you add? You have to come here. You have to support me. Yeah. Okay.
I just received an emergency message.
Yeah. Okay. Whether there's a possibility, you give one mic to him. Yeah, you can use this.
Yes, so sure, it will be a possibility. Maybe the one thing that I'm thinking of that could maybe hinder a little bit this kind of programs is that in France, most of the teaching is done in French.
Yeah,
yes,
okay.
that's and we don't have maybe so much international programs so far. Right. So, but we have to develop this to my point of view. So, yes, so to me it will be maybe some specific programs or maybe some specific courses that that could be done with this. Yes.
So this program, when we introduced for University of Dalhousie with TNAU, it was English speaking. Both the courses are English, so language, medium of instruction is English. So we could do that, but in your case, it's not possible. So just we'll keep it on hold for the time, because undergraduate level, it's not possible because their medium of instruction is French.
Yeah, but it depends. So first information, I'm not aware of any course in Tamil in France. It's an important information.
Okay, right.
Second, we are connected to other schools because Agro Rennes or l'Institut Agro is a big school in France. But there are other schools that could be contacted, because I know that in Toulouse, for instance, there is a private school who organized a double degree, or at least a common education program with India. So I know it's already existed.
Right. Okay.
So of course the language is English in that case. Okay. So I don't know if for this French delegation we should try to open the collaboration to other schools that also like AgroParisTech or ENSAT in Toulouse or whatever, Isara, Lyon, whatever.
So the degree program may not be possible. At the same time, short-term visit possible. Because there is a program called IDP, Institute Development Program, where the students supported by Government of India, they identify certain lab, the students go there for a month and come back. So that type of arrangements can be done. So undergraduate level, degree not possible, but there is a possibility that the students can visit, learn something and come back, and for which a certification can be given.
Yes. For internships, for example, like this.
Okay.
Yes.
This is fine. TNA folks, all of you agree with this?
No, it is exchange.
No, no.
They will also come here or
No,
not?
no. Naturally, US Francophones, they can also
of course, yes.
Can come here also, right? So it could be exchange, but the exchange, how it is happening in IDP program, for international program, the students are funded by Government of India and they are going to other country, no bench fees, only the accommodation as well some other logistics are covered, no bench fees thing. So that can be explored, okay? In case US students want to come here, there is a possibility, we can explore the possibility. That is number one, correct? Undergraduate level. The number two is master degree. In master degree, TNAU will be very much interested to spend one semester in any of the academic institution like the here, research organization like Institute for Agro, RENIS, Agro, like INDRA earlier. So that is possible. One semester, do research in academic, research organization, and the professors here in TNAU or professors in other research institute can collaborate and do the research together. Is it possible?
Yes, of course, this can be possible, and also for the teaching courses, we have, for example, one master degree in seed seed production and seed quality that is entirely in English. So yes, we can also welcome students for these specific courses.
Research.
Yes.
So for master's research, the students can spend one semester in France Institute, any of the France institutes, for which research collaborates. Of course, I cannot speak for TNAU at this moment, but certainly I inform acting Vice Chancellor to take it forward. So that is possible. Okay, that is for the master degree, correct?
Yeah, correct. So then there was the first answer was for education, from what I understood, and yes. I'm thinking about internship. So for internship in RAE, I don't speak about CIRAD, but I guess it's the same, and Sébastien for R&D.
At what level? What level?
Yeah, master, for 6 months internship.
Right.
I think it could be possible to have supervision or joint supervision for a 6 months topic for a master's internship. I think it's not a problem at all because we often hire at this level.
One condition is that internship must end with what? High impact factor publication. The students must get a publication in which both TNA professor or ICR professor as well from your institute can jointly publish a paper.
Yeah, in France it's possible, but it's not mandatory. It's mandatory for PhDs.
We have mandatory.
Okay, so we have to handle the difference.
Yes.
Okay.
Okay, is it clear? Master degree is fine. Next is a PhD. PhD is a 3-year program. 2 years the students spend on research. Certainly this program will be very helpful. So undergraduate we have decided, master degree is over, and PhD level, there are some common interests. For example, somebody was speaking about wheat rust, which is a very global issue. We have techniques, we don't have answers. So that kind of a thing can be collaborated. So what we do, we are PhD also, at least 2 semesters. 1 year they have to spend the time in France, same thing, your students come to here. Mutual exchange is possible. How it is going to work? We have to look at the modalities with the administration of TNAU. I cannot speak at this point because there are lots of issues involved. There is academic council has to approve, and how many students can go, what students can bring, those things have to be decided. Again, for PhD, at least 2 to 3 papers, high impact factor, publication is very important. TNAU focus on publication because that is mandatory for the students to publish, right? So that way we can help. So education is over now, right? Correct? Everything is fine? So within the education, suppose there is a demand for a particular graduate. I am talking about employment. Is it possible? Indian TNAU students get employment in France at the undergraduate level or PG level or PhD level?
Yeah.
You don't need to answer now.
No, I can answer because it's very competitive if they want to be hired as a tenure researcher.
Okay.
It's feasible. For instance, I don't know, I have to count, maybe 8 years ago, we got a great colleague coming from Nepal. But it's very rare. After, what is very feasible is postdoc. It's easier in terms of probability of being hired. And after, once you get, because like in India, you have to get used to the system. Because if you're coming right from India trying to make the competition, no chance at all. You have to understand the way it works in
postdoc is possible.
Yeah.
That depends on the individual's caliber, individual strength, scientific sense, publication, communication, the type of research. It's all individual based. Okay. So we decided undergraduate, master's, PhD, and postdoc. It's all done. Education is over.
Yeah. Just one remark. I can talk about my institute. If we, for PhDs, we are able to receive from any country in the world PhD student. That's okay. However, we cannot send in RAE. We cannot send our students. When I say ours, is people who are paid by our institute abroad. We don't know how to deal with that. I had last year a student who finished her PhD. She worked with Cambodia.
Okay.
But it's not my institute who hired her. It was CIRAD, because they know how to put people everywhere around the world.
Okay.
So if you want that type of collaboration, I think IRD or CIRAD are more used than in RAE to send people abroad. Yeah.
Okay, right.
Yeah, Thierry wants to add something?
All right. Certainly, whatever I mentioned, this is for discussion. Certainly, Sendil Raja has to make a note of it, make a proceedings, submit to Tamil Nadu Agriculture University, and the administration has to agree on, and we'll take it forward. I already mentioned the acting vice chancellor. I hope it will work. Okay, so these are the then modalities. Once we decided this, who will pay for the students to travel, how accommodation is done, those things we'll do it later. Right now, there is a possibility for master's student, PhD student, go for internship, get some publication done. That is possible. The modalities had to be evolved. Okay, it's just correct number of students. What is the cost involved? Those things had to be decided. Okay.
There is another option that you didn't mention related to education. It's online teaching.
Online.
Because I think
you make a note of the ODL, or open distance learning.
Yeah, because in your organization or even myself, we designed training courses that are available in English with online exercises, feedback, and everything that can be self-explaining for students. So it's another option.
Tamil Nadu Agriculture University has a directorate for open and distance learning, where regardless of their background knowledge, they can register to a particular program and learn. So that type of online teaching is possible. That can also be noted. And if some students interested to have online courses taken from France, it is fine. It is possible.
Yeah. And there is also something at short time. It's what I mentioned. It's the organization of a summer school that will be focusing on new researchers. either PhD students or young researcher, or why not older researcher who wants to focus on a given topic. And that can be nice because we can organize it with Indian teachers, French teachers. So we would have to work on the program, but I think it's quite feasible.
Right,
it's not infeasible, but it's another option. Yeah.
Right, please.
And maybe what we can do also is open like a channel of communication. Maybe if we have one person that is dedicated, for example, to diffuse the information, if we have something that is open internationally, we can send to this person. Maybe it could be Sentiraj, maybe it could be another person, but it could help.
Yeah, sure.
Yes.
In terms of course, the education program is concerned, we need to get the consent from the students. Students should be capable. Students should be interested, parents should allow them to go. There's other criteria because I dealt with Cornell University dual degree program long back. Some students, find it very expensive for them to fly and all. So we'll look at it. So as of now, we have taken different options. We'll just explore how it can be done. The ODL program is easy to do. Summer school, it is possible. Those things can be done. So education program, everything is done. Okay? All of you clear? Any points from students? I'm speaking for you. Is there anything?
Would you like to follow lessons with a French accent?
French accent? No? Clear? Rendu paper nalu paper eludano. Sorry, I'm speaking in Tamil. You have to write 4 papers a year. Future. Right. Then another advantage of this type of program is that the government of India has introduced a program called National Education Policy. According to National Education Policy, this is encouraged. So another few years from now, our state government also has a policy that the students are encouraged to go out, encouraged to learn many things. So I am also involved in certain government programs. So certainly, whatever we discuss, it is useful, doable. Okay, done. Education is done. The second part is the research. The second part, research, how it can be done. We have TNAU, Tamil Nadu Agriculture University. We have ICR Institute. We have research scientists. We have scientists from other universities. So each one of them may have different interests and all. But for a major part of agriculture research, TNAU should be point of contact. The TNAU would be the point of contact. So according to me, because I am involved in various committees, so research program, joint program application, it depends on the initiative of a particular scientist and the interest of the scientist from France, and have a collaboration regardless of organization, because that is open. Okay, is it clear? Because we have Bharatiyar University, we have ICRISAT, we have ICR, because everyone, Darward. So we have a different scientists from different organization. We cannot combine this. TNAU scientists, if they are interested to take it forward, that is fine. But if somebody interested to initiate something, this particular platform would have helped them to bridge. Correct? Am I correct? Okay. Right. So this is the way the research could be done. But any point here?
Yeah, I would like to say a few words about the funding. In France, one important source of funding of research projects is the European Commission.
Okay.
So we have to gather a group of maximum, let's say, 20 countries, and we can get several millions projects. So it's nice. During the last 10-15 years, I coordinated several European projects. And now it's more and more open to countries outside of Europe, such as China, Africa. And I don't know, I asked a question to my French colleague, do you know if there is any opening towards India? I didn't see that in any call at the European level, but maybe we can check if this is happening, because there is the will to strengthen the link between France and therefore maybe Europe and India. So that would be one way of potential funding. The other way is that for each of our French research institutes, such as IRD, CIRAD, and INRAE, and I don't know about your institute, sorry, we have international division that supervise the cooperation by countries. So we can ask to our colleagues what are the specific funding towards India. So I'm sure that at least there are people and they are aware of, and I think the Ambassade de France, and also there are what's the name, la fondation qui... Well, l'AFD, the French Agency for Development. So they can provide a lot of funding. So CIRAD is used to work with them a lot. And there is also an initiative from you, CIRAD, about pesticide reduction. And it worked for many years, and now it's a new program. François Cotte and so on. But anyway, it's a specific program for pesticide reduction in the tropical area. Okay. So that can be also something. Okay. Then there is another initiative worldwide. It's called ACP. So it's an initiative for agroecological crop protection. So I mentioned it. There are Indian colleagues involved in that, but they are up north of India. Okay. But I think it's not a problem to include other partners, I guess. So
this research funding for individual project depends on the strength of the scientist, strength of the other side of the scientist, strength of your proposal. It is you are defending a proposal, like a DBT, DST. You defend a proposal as the Indo, French program, immersion program you are doing, and do that. Okay. So he suggested few things. Of course, Government of India under DBT, DST, there is a program called India-French program or there. You apply, we'll help on this. Okay. So this is an individual scientist program. The next research program would be at least one strong program must be discussed and developed, conceptualized, developed, delivered for TNAU. What is that you need to have a discussion separately, because that would be a major project. So you know the strength of TNAU, you know there are lots of strength. So your team can also have a discussion and develop a major project. So that project, I don't know how we mutually fund or who will fund, you can evolve a good project. So again, the point of contact for TNAU would be Sendil Raja for us, I think. Then we can minute it and it has to be evolved. So it needs lots of things, and multi-institutional can be done, because ICR has germplasm exchange and all, ICR is involved. So ICR, TNAU, French organization, all the things together develop as a major project. It would be a 5-year project, 10-year project. Some of the challenging diseases like gingerly phyllodes, some of the challenging diseases like smut disease, which earlier it was referred as a minor, now it is major. So that type of program we introduce that. Doctor, you are agreeing with this pathologist? Yes. Okay. All right. That would be the major project. It has to be discussed. It could be multi-institutional. It could be discussed technically, because we have to evolve a program and what to be done. So according to the Government of India program, there's One Health. That is a concept Government of India has introduced. So under the banner of One Health, we can put all the ingredients together. Right? So that can be discussed. This is a major project we can think of. Right? So individual project, institutional project can be done. TNAU can do many things. Nano they can do bioinformatics, they can do system biology, they can do molecular modeling, anything you name it, TNAU has expertise. So that type of project can be done. So I can facilitate to some extent to do that. Is it clear? Is it clear, Doctor? Okay. Individual project.
Just one question to have the order of magnitude. How many researchers do you have in TNAU?
In total, we have 950 scientists as a whole. But if you take pathology, it may be 75? 78 pathologists. But we can integrate. We have nanoscientists, we have molecular modeling specialists. And when we develop a multi-institutional project, because I handled a project where 7 countries were involved. So I have to manage all the 7 countries speaking different language, different culture. We have to manage it. So in this particular case, we identify particular area. I think from morning I am watching some of the serious matter like phyllodes, what Sendil Raja planned to do. So you have to put many things together to target particular disease, mycoplasma disease or something, and you use molecular tool, you use statistical tool, you use nanotechnology, you use diagnostic tool, everything together, develop some robust thing that can be done. So that would be very interesting for us. This is the example. That doesn't mean that I am targeting Sundararaja to promote, but it's an example I am saying. It can be done. Okay? So we have decided individual project, we have decided institutional project. Is it clear? Okay. What else can be done in research? Please. Okay. Under research, scientist exchange has to be done. Yes. For example, the Canadian project, 18 scientists, I took them Canada, United States, many countries. We went to Cambodia. So likewise, this particular platform can help in visiting at least France or EU countries at least, like Netherlands or few countries nearby. So, you make a note of it because this involves money. I cannot speak for any of the institute at this point, but it involves money, involves administrative framework. So I cannot speak at this point, but faculty exchange is possible.
Yeah, I think we talked a little bit about that. I know, for instance, in my area in Toulouse, we have a program where we can invite, usually it's a senior professor who has to teach, not so many hours, but to teach to the associated Institute for Education. And it can be up to 1 month. And it's quite nice. If it's possible to write a joint paper, it's perfect. Okay. And I think that in Institut Agro, you also have that type of
we
program.
As a sabbatical. Yeah. You are referring to sabbatical?
Yeah, but short time, up to 1 month.
No, TNA can afford more than that. In other words, it's a short sabbatical, 1 month maximum. I think TNA would be very much interested to send faculty abroad for a period of 1 month with a specific line of activity, writing papers, visiting institute, develop a conceptualized project and all. That is possible.
But the call is only once per year. You cannot apply when you want. There is a rhythm.
Yeah.
There is maybe 1 month or 2 months where you can apply, and after you have to wait for the next year.
Yeah, right. So how many, is there any number of scientists could be recommended for the particular slot?
Yeah, in our case, it's usually one.
Only one?
Only one, yeah. But I mean one per research unit.
Yeah, these are very important, Doctor. In scientific community, you have to have your own strength, your own managing things. So it's individual strength is very important. So anyway, we've opened up an opportunity for TNA scientists to go abroad, maybe 1 or 2 or 5 max 1 year.
Yeah, because just to give you an order of magnitude, in my INRAE center in Toulouse, it's about 1,200 researchers. Okay. So it's quite big, if you compare to the size of this, and it's only a small part of the entire system in France.
Oh, great.
And so in Institut Agro, I don't know how many teachers and researchers you are.
200? Yeah, 100 in Rennes, 100 in Angers, I would say. Yeah.
Actually, we evolve a system. Any program that is introduced in Tamil Nadu Agriculture University, it has to be approved by 2 levels of approval. That's called Academic Council, then Board of Management, that University Vice Chancellor take a call on that, take a decision and develop a framework. Accordingly, it will work. Okay, it will be open to all. Though it is discussed for pathology, it will be open to all only. So that can be done. So that's a research part, no problem, it can be done. Yes?
We're not working on the different institutes, different places. So like there is maybe only one possibility in the laboratory of Jean-Noël. There are also some possibilities in my laboratory.
Yeah, sure.
So it's
we will welcome
to apply
you.
In different places.
Where is your lab? We want to come. You want to come right away. Right. See, the possibilities are there. We all having few contact points. Each contact point has its own strength, right? So it depends on mutual interest. We can evolve and use it, right? Research side we can use it. Is it clear? So now we decided individual research, institutional research has been done. The institutional research needs lots of time, conceptualization, discussion, brainstorming, and the director of research, vice chancellor, whoever it may be, they have to make a decision. Good project, like a Canadian project, I feel proud of. I got 12 crore Indian rupee, which is about 2.5 million American dollar I got from one source. So that's a big project. Likewise, somebody can be proud of getting that much of, it's a global funding. So that type of thing can be done. So that is possible, okay. The third aspect would be, how do we enhance or how do we meet next time, this particular event? Now we have conducted this event for 2025. Will it be 2026 there will be event? Yes? You are proposing. What is your concept then? Okay. Okay. So what we do, because we are very close to you now, we begin to understand your strength, you understand our strength. We had a very good time in a Marriott hotel. Certainly we can think of annual event. This event would be made annual. Can it be done? Who is funding this event? Okay. It is Indian government funding. Okay. Indian government funding. We are funding your okay, right. Thanks to Prime Minister Modi. Okay. Right. Okay. Okay. But you got. All right. No. This all arrangements you are paying from the fund you got it from DST, na? That is Indian funding. Okay. Okay.
Yeah. Can we... I think with that, I think we have two options. Either it's something that can be organized on a yearly basis, trying to meet and so on. But maybe it's not the most efficient way. Maybe the other idea could be to say, okay, no, the next time it's something concrete that you do. Either it's an international conference, either it's a summer school, either it's a training session within your university.
Senthil Raja, because you demonstrated year one very effectively, you continue this, try to get funding every year. Alternatively, at least one international conference can be organized. One international conference involving scientists from both the sides, maybe other scientists as well, with a focus. Not too many people. I think I am very happy with the crowd here, because many of the TNAU conferences, now nobody is there. Nobody is there. I speak for empty chairs, but here is very effectively done. So what we do, international conference can be done. Alternatively. So international conference can be organized with a specific purpose, specific title, theme areas. All the participants must attend. No deviation in that. Okay, it can be done. Okay, right. What else you want to discuss?
Some kind of technology transfer
okay.
possible, and also farmers visit for the benefit of the farmers. Even Tamil Nadu government now they are allocating some budget.
Okay, right. There is another last part is the extension. Last part is the extension. It's a social science. So Tamil Nadu government has money for the farmers to visit some places. We'll pay for it, but we need a facilitator. Tamil Nadu Agriculture University have MOU, will establish MOU with you. Then we are married together. We will facilitate their visit to your place. That's what you wanted, right? So that is, we are helping the government of Tamil Nadu to do that. So again, it is a exchange of farmers visiting grapes area, vegetables area, millets.
Yeah, for this there are many options because our institute here are not really involved into extension. Okay. In France, it's again another organization, and we have Extension Institute. Okay. And with that, we have very good connection to experimental platform or even network of farmers. Okay. And I think about our national action plan, we have 2,000 farms that can be used to present to Indian farmers what's going on in France. But we could also imagine going to a bit closer in the Indian Ocean because there are more tropical production. And that could also be very interesting, or maybe in the French Indies. I know a little bit less this territory, but We saw that the chocho, the bananas, and we have many tropical crops in common.
Okay, the last part would be technology and the genetic resources exchange and all. That is a very ticklish issue because NPPGR and all, they know very, very difficult process. I don't want to get into problem. But in general, some of the technologies can be developed together. Some, you may be expert, your scientists may be expert in certain areas, maybe diagnostic kit, maybe, molecular modeling, maybe bioinformatic tool, maybe nanomatics or something that we can do. We can develop technology together. That is possible, right? The professor, scientist from ICR, he was asking about NPP material exchange, right? That is a ticklish issue, right? There is a NPPGR, ICR has to sign MOU and all, right? To have resistant source, we get it from other institutes. So that you deal with separately. But if any exchanges involving TNAU, involving ICR Institute, we can club together. Okay?
Yeah. I think this can be possible, but we have two warnings. The first one is IP issues.
Yeah, again.
So it should be clear from the very beginning. So that's why it can be, if we go into that, we should work together to write down.
Exchange, we will be cautious on that. Because
so this is one thing.
Certain resources we cannot, as many, even they don't want to reveal the name of the disease.
And the second thing is the DMP, data management plan.
Okay.
That is also very important if we share data sets and so on.
I think we have discussed everything. Everything all together. Yes, you have a point? Huh? You speak. Okay. Yeah, I just read what Dr. Kumar, who left, has pointed. There are 5 points he suggested. Collaboration, he is from the ICR, Indian Council of Agricultural Research. It's a national research system. Collaboration on epidemiology, ecological plant pathology, experts available in France, Professor Sergei Savary and others can be linked. That is number 1. Number 2, plant microbe for foliar disease management in wheat and other high value crops. That's a point 2. Point 3, NLR discovery for biotrophic pathogen management. 4th, pathogenomic on Xanthomonas. Xanthomonas is a serious problem. Xanthomonas is a bacteria. Okay? Then 5th one is a sandwich program that I already discussed. Okay, because he mentioned it is my duty to deliver in front of everybody to make the system, everybody knows what we are speaking about. To initiate everything, there should be one initiative had to be done immediately. That is MOU. I think MOU, right now we don't have MOU with TNA, right? MOU, Memorandum of Understanding.
We have an old MOU from 2003 between ICIR, INRAE, and CIRAD.
Okay.
And I know that they are progressing on for a new document based on that, but I don't know when it will be formalized. So the feedback I got from the
ICR.
International Division of INRAE is that we should use this document as a general umbrella rather than making MOU with individual universities. And also the advice should be to write down another type of document, which is letter of intent.
Letter of intent, okay.
But also that type of document involves some legal work and coordination within France and with Indian institution, and also some administrative work. So it will not be done tomorrow, because tomorrow is Saturday, but, but it takes some a little bit of time and coordination.
For all the process to get started, there should be a starting point. There may be, the final point would be for Sendil Raja would be MOU to be established, right? That is the last point. The last point would be the first point, because MOU to be established, then only other things will happen. Okay, so what we do, there are two levels. ICR, they are also friends for us. There's a national level. In state level, we have Tamil Nadu Agriculture University. Agriculture being the state subject, we deal with the state administration. So the university vice chancellor take a decision, major part of decision is taken by itself. So there will be a general MOU indicating these are the institutes in France. Tamil Nadu Agriculture University have MOU to have a joint education, research, extension, student exchange, all together it becomes one general MOU. Then specific cases come later. Again, to establish TNAU to that document, it has to be approved by ICR, Indian Council of Agriculture Research. That TNA administration will take care of it. So we'll try to do that.
Yeah, I think what we need to do is first, at least I speak for INRAE, to work on that topic. And already for one institute, it's not so easy. And after, try to coordinate the legal bodies of each of the 4 institutes involved in this seminar, and see, because the problem is that the MOU, the existing MOU, does not include the Institute Agro-Ren and IRD.
Okay. But in case of Tamil Nadu Agriculture University, I know TNA administration very well. I was director of research for 3.5 years. I know TNA administration is compared to national institutes. TNA administration is somewhat simpler. So we can list what are the institutes, what are the strength. So we develop a MOU indicating all the ingredients. Make sure that all are signed on same board. Okay, we can do that. Suppose if you want to split organization 1, 2 like that, do you want like that? Do you want one institute, second institute, third institute, same content?
Sir, like our ICR system, do they have any federal organization?
No. That is why Monsieur Noel is very important. He is the person. Yeah, all national level only. See, in case of what I understand from their conversation, in France, the research organizations are national. Correct? National. Like Canada, national level organization only. The universities, there is no university exists in agriculture. It will be colleges or universities. And they will be facilitating us to have a link with them. Correct?
Yeah, we are used to work together.
Okay.
I don't say it's easy, but we always collaborate, and we are even integrated within what we call mixed research unit, combining education and research.
Yeah.
So that's why it's not a problem, and we have many contracts with CIRAD, a little bit less with IRD, sorry Sebastian, but we do have collaboration, of course.
No, what do you think? You want education separate MOU, research separate MOU, like that you wanted? Or you put all the things together, all the organization together, make one?
No, I think we should go for the simplest solution, because it will be
complicated.
So the aim should be to simplify everything, because I think also maybe it's easier from your side because you are only one university and it's one administrative body. Within INRAE, it's already very complicated. You know, it's very administrative. We have many layers of decisions.
Right.
And after, when it comes to coordination,
you suggested, because when we minute it, whatever you are saying, it will go as a record. So what you do, which is the simplest way we can handle? When we handle one institute, this education program like that, it will be easier?
Yeah, I think so. Yeah.
Okay. So we identify education, what could be done, research, what could be done, or social research, what could be done, and other ODL program and other things, what could be done. So it's all from you only. So we identify to whom I should talk to education among the 5. Education, both of them. For research?
Mostly topical.
4. Then for social research, connecting. Okay. So, okay, we'll go with the simpler one. Okay. You got the point, Doctor? All right. Okay. So these are the things we do, and
There is another category of actors that you didn't mention, and who are key players in agriculture in India and in France, is the policymakers.
Yeah.
And I don't know, I could, for instance, try to motivate the French representative of several ministries to come as a delegation and to visit, and also with an educational decision at the ministry level, or on the contrary, to have feedback from your national policies.
In terms of university administration, the policy making body is the vice chancellor for university. Okay, because why the vice chancellor is given superpower here? Because once the decision is done by university, that also includes the state government, whatever the regulations. It also includes ICR into consideration. So the administration, TNA administration, or it may be, so that individual will take a call on that and make a decision how to make it simple and go ahead.
Yeah, because there are some processes that are very important. For instance, how you register a new pesticide for the market.
Okay.
In France, we have specific national bodies linked to European bodies, and there is this process. I don't know at all what is the process in India.
Okay.
And for instance, when I ask the question...
That policy is a very complicated policy here,
Yeah.
because to register a pesticide, it is a long process. We have a national level apex body called CBIRC. That is a organization. Yesterday I had a meeting with them. So that is the apex body does registration of pesticides. As an individual TNAU administration, we will not be able to register a pesticide because it costs so much.
Yeah, but what I had in mind is not maybe doing the thing yourself, is just making the bridge with a proper actor, and that would be also it would be an outcome
At the
of the seminar.
At the same time, this is for pesticide. At the same time, for nanotechnology, because we are very leader in nanotech, we developed the policy, we developed the guideline, we regulated the rules and regulation. Everything was dictated by us. Whatever we have said, TNAU, whatever said, this is the rule of the land now. So for nano products evaluation, and the TNA is the authority. TNA lab is the referral lab.
Yeah. So for instance, if you're interested, we could try to identify the actors who makes the policies at the national and at the European level for the nano technologies, if you're interested, because I see that there could be mutual benefits from knowing how it's done in the other country.
Yeah, correct. Particularly nano products, of course, I know the global scenario for nano product. European Union is very stringent. They don't want nano. The word nano, they do love to hate it. They don't want that. Okay, to some extent they agree, like US, Canada, China, Australia, they all agree, except
Yeah, and with regard to policymakers, the third version of the National Action Plan for microbiome agent was very efficient in France. And I see that all the problem you have with that, not only for plant diseases, but also for human health and animal health, I'm sure it would be very efficient for the Indian government to try to improve the national policy to reduce the risk.
The national policy for microbiome has been done already. It's a very recent one. That is national policy on microbiome. Also that biostimulant. We did not have regulations earlier. Now the regulation in place. Few products have been registered. As you said, in the event of residue, event of toxicity, pesticide reduction, this is an alternative. So that is in place in India as well. So that is a policy, it's going on in parallel. So these are the points we do. I think we'll chart out all the points we'll discuss and get back to you. Once we agree, it can be submitted to TNA administration. When it's corrected, so we'll take forward from here. So are there any other organization want to speak? ICRISAT, ICR, because KAU, right? It's all individual project only, right? Or you want to say something? Like, you don't want to? Okay. Agree, sir. Okay, did I moderate well? Okay. Okay, you want to say something, Doctor? You just share your thought.
Yeah, thank you. So I think you made our job easy. So your conversation is so
No,
deep.
I have to thank him because
Yeah.
he only narrated these are the areas can be done.
Yeah.
I took a cue from him and proceeded.
Thank you, thank you. So I will take you all apart from this collaboration to the workshop again. So what we have been discussed the last 3 days, 2.5 days. So I would like to recommend, I hope you all agree or you may disagree. This conference or seminar discussion, the presentation, what we have made so far, can be made available and accessible to all the participants is the number one. I feel that is most important as a researcher. Is all the time? No. So what I mean to say, the slides, okay. The slides is accessible to all the people. You can still consult with the individual presenter. If anything is a confidential, we can remove and you can reshare it among the groups. That's how we community of practice, we do all the seminars. Number 2 is that outcome of the recommendation and discussion in each session, and also each of the presenter can be come out as a finding or recommendation out of this workshop. So I think if we need to deep dive into those is very critical. That also help your further collaboration and identification of what is a priority area, either in the state or across the state. So that is very important. And third thing is the most important, as everybody's talk about the documentation and publication, including Professor he pointed out. So why don't you think this group discussion and this area, what is the topic itself, interface and under the One Health umbrella, why don't you develop a discussion and white paper and based on the topic discussed in the last 3 days and circulate among them and fix the tentative deadline to contribute all of them, and you can submit, you can be a document that should be shared to our funder, donor. The shortest version can go as a journal publication. Yeah. So that is a very critical part. So that's what you can build on our wall, further, deeper or longer.
You got his point. The point he raised is very good. Is it possible?
Is there already the plan?
Okay.
Journal.
That you can. No, they can discuss.
We are talking about impact factor.
Tell me the impact factor.
6.
Above 6.
You want to leave? Okay. So, okay. I'm almost done. I think you decide as an organizer and co-organizer. Please, you decide what journal. But these are all my point. And last but not least, identify at least some priority crops to start, as a pilot work, and also which disease you want to start with that. And also think of building a core team, which comprises the multidisciplinary team, which drive, as you see the blue sky research, because this is kind of the blue sky research, not the ordinary. So if you want to build the blue sky research, we need to have the diversified expert in those umbrella. It's not just One Health is a broad umbrella, but we plant health, bring it to the plant health. So how we can identify those 2-3 crops relevance to, maybe higher relevance to India, maybe somewhat least relevance to France today, but what would be the future for the France? Like that. Thank you.
Yeah. Okay. The final point you noted is the publication of the papers. Publication.
Maybe we can start right away. Can you host 2 of us? Because we will miss our plane and we have to stay with Nicolas. No, sorry. We really have to rush because we might miss our flight.
No. That is why I did not allow the audience to speak much.
So
I
please-
spoke everything what they
No,
think
so,
that it is
so please you can keep talking, and thank you very much
Okay, thank you.
for the conclusion and the nice session.
Very good, Noel. Very good.
So we are sorry, but you still have 4 Frenchies to discuss with. Okay.
Though I am with the industry, but my heart is with the...
Okay, so thank you very much once again to Frédéric and to Senthil Raja. Salut.
Okay.